All right. Hi, class. Today in chapter 13, we're going to focus on carbohydrates. This chapter is going to have a little bit of a different feel than the last few chapters. So what I've done is I've taken a snapshot of your information sheet so we can go over kind of our game plan here. If you focus on the bottom right-hand corner, that's where we have for you a summary of the functional groups from the organic molecules that we're going to go through this term. So what we've gone through is we've talked about hydrocarbons, which are the alkanes, alkenes, alkynes, and aromatics. We've also talked about the alcohols, ether, aldehydes, and ketones. What we're going to do today is we're going to take a break from organic chemistry, and I'm going to talk about carbohydrates, which falls into the category of biochemistry. Biochemistry focuses on molecules and reactions that are going on in living organisms. So after this chapter, we'll come back and I'll pick up again with carboxylic acids. So what are carbohydrates? Carbohydrates are our major source of energy from our diet. They are composed of the elements carbon, hydrogen, and oxygen. They are also called saccharides, which means sugar. So I do want you to know that you can interchange those words. So I want you to know that a carbohydrate is also called a saccharide, and it is also called your sugars. So where do we get carbohydrates? They are produced through photosynthesis in the plants. Carbohydrates such as glucose are synthesized in the plants from carbon dioxide and water. So I think we kind of have an idea of this, that the plants take in carbon dioxide from the air, and then they take in energy from the sun. And they use that carbon dioxide and energy and also water that they take up through the roots, and they use those to synthesize glucose. Carbohydrates are then oxidized in the living cells, which produce carbon dioxide in the water, and then they also give us energy. So it's the opposite of the reaction that's happening with photosynthesis. Now there's different types of carbohydrates. There are monosaccharides, which are the simplest carbohydrate. So that's the smallest unit of a carbohydrate you can have, is the monosaccharide. Remember, the prefix mono means one. And then there's also what they call disaccharides, which consist of two monosaccharides bonded together. So remember, your prefix di means two. And then I'm going to take you through polysaccharides, which poly means many. So polysaccharides contain many monosaccharides bonded together. Okay, so if we look at this picture, we have talked about, actually we haven't gotten into that reaction yet. When we get into carboxylic acids and ethers and amides, I'm going to talk about a reaction called hydrolysis, which is when you break apart molecules using water. So if you take a monosaccharide and you add water to it, nothing's going to happen. You're not going to get any hydrolysis because water's not going to break apart a monosaccharide. But if you have a disaccharide and you hydrolysize it in the presence of an acid, we'll go over this a little bit more in detail. Hydrolysis will break apart your disaccharide and you'll be left with two monosaccharides. If you have your polysaccharide and you hydrolyze it, you are going to get many monosaccharides. Okay, so focusing on monosaccharides, what I'm going to do is I'm going to take us through three different types of monosaccharides. There's many out there in nature, but I just want you to get a taste and a flavor for what monosaccharides are. So I am going to want you to memorize the name of these three and where we find them in nature and things like that. So what are monosaccharides? Well, they consist of 3 to 6 carbon atoms, typically. They have a carbonyl group. So remember, the carbonyl group is the carbon double bonded to the oxygen, which means they're either going to have an aldehyde or a ketone functional group on them. They have several hydroxyl groups. So do you remember what a hydroxyl group is? That is the oxygen and the hydrogen bonded together. And carbohydrates are broken up into either being an aldose or a ketose, depending upon if they have an aldehyde or a ketone. So aldoses are monosaccharides with your aldehyde group. So here is the functional group aldehyde. We've covered this in the last couple slides in organic chemistry. So you want to be able to identify this as your aldehyde group. It's got the carbonyl group, and this carbon is bonded to a hydrogen, and it's also bonded to a carbon. So we call these aldoses. The last 3 letters in the naming of almost any molecule in chemistry gives you a good idea of what class it's in. And your carbohydrates are going to end with OSE. So that's why they call this an aldose. It's letting you know it's a sugar with an aldehyde group. So aldoses are monosaccharides with many hydroxyl group. And then you can use a prefix to let the reader know how many carbons there are. So a triose is a sugar with 3 carbons. A tetrose would have 4 carbons, and etc. So it's just kind of another way of grouping them. Actually, let me go back to this slide. So this particular aldose is an aldotetrose because it has the 4 carbons in it. So you'll come across aldo, trioses or aldo pentoses, depending upon how many carbons. So the aldo in the front lets you know it's an aldehyde group. The tetra, whatever prefix is here, lets you know how many carbons, and the ending OSE lets you know you're talking about a monosaccharide. Ketoses, on the other hand, are going to have a ketone group. So let's find the carbonyl group here, right here. And this carbon is bonded to a carbon on one side and a carbon on the other. Notice in biochemistry, they start to write their carbons vertically instead of like organic chemistry where we write them from left to right. But it's just the same molecule, just written top to bottom. Ketoses also have many hydroxyl groups. This is going to be common for all of your carbohydrates. Fisher projections, we talked a little bit about this in the last chapter. A Fisher projection is used to represent carbohydrates. Remember, we talked about it when I was teaching you stereoisomers. And you use a Fisher projection when you've got a chiral carbon. So here it is again. We talked a little bit about this already. And all of your carbohydrates are going to have some chiral carbons. So Fisher projections are used often. So we use them to represent the carbohydrates. The way we do it is we place the most oxidized group at the top. So that's kind of the general way of doing a Fisher projection of a carbohydrate. Another way to say that is you write it so that your carbonyl group is as close to the top as you can make it, because that'll be your most oxidized group. The Fisher projections show your chiral carbons at the intersection of your vertical and horizontal lines. So in a Fisher projection, the hydroxyl group on the chiral carbon furthest from the carbonyl group. So you find your chiral carbon that is as far away from that carbonyl group as you can get, and that'll be the carbon that is going to help you decide whether it's a D or an L isomer. Okay. Another way of saying that is locate the crosshair, your chiral carbon that's nearest the bottom. Okay, because you've put your carbonyl group as close to the top as you can. So find the crosshair nearest the bottom. If your OH group is on the left, then that's going to be the L designation. If the OH group's on the right, then that is the D. Once again, remember that isomers, these D and L are stereoisomers, so they are mirror images of each other that are different molecules. So some examples of D and L isomers are monosaccharides. Here's glucose. I am never going to ask you to draw glucose or be able to recognize this as glucose. Okay, so don't panic about needing to memorize these formulas and identify what monosaccharide it is. But I am going to give you a monosaccharide and ask you, is this the L form of it or the D form? So this particular one is glucose, and we find the crosshair nearest the bottom, and the hydroxyl group's on the right, so it is the D isomer. Here's ribose, and once again the OH is on the right, so it's a D. If we look at galactose here, it's a different monosaccharide. It is in the L form because this hydroxyl group's on the left. So it turns out that only the D forms of your carbohydrates are the ones that are abundant in nature. So most of the carbohydrates that we metabolize are going to be the D forms. So here's a quick learning check. Identify each of the following monosaccharides as being either D or L isomers. Okay, I hope you all here said, here's the hydroxyl group we're looking at. Since it's on the left, this is going to be an L form. Here the hydroxyl group is on the left, so once again this is going to be L. Okay, and remember we are looking for our lowest crosshair. So we first focus on this particular chiral carbon, and then we find the hydroxyl group on that. Since this one's on the right, this is going to be D-fructose. Okay, so I'm going to take us through 3 monosaccharides. The first one is going to be glucose, and I want you to memorize some of these facts about glucose. For starters, glucose is found in nature in a lot of our fruits, corn syrups, and honey. These are just some examples. It's found in lots of things. It is also called dextrose, so I do want you to know that, and it's also called blood sugar, so it has those 3 different names. It is a monosaccharide building block of starch, cellulose, and glycogen. This particular bullet is going to be easier to remember after I've talked about the polysaccharides. So starch, cellulose, and glycogen are polysaccharides. We're going to end this chapter with them, and you're going to hear me say over and over again that the monosaccharide that makes them up is the D-glucose. Okay, the second monosaccharide we're going to focus on is fructose. It happens to be a ketohexose. So let's make sure we can identify that. Why is that? Well, here's the carbonyl group, and it's bonded to 1 carbon on one side and a carbon on the other. So that's what makes it a ketone. And then if we count our carbons, 1, 2, 3, 4, 5, 6, there's 6 of them. So the keto means it's a ketone, the hex means there's 6 carbons, and the ose means it's a carbohydrate. So that's fructose. It happens to be the sweetest of the carbohydrates found in nature. It's found in fruits, fruit juices, and honey. Once again, it's found in lots of things. These are just some examples where you find them. Fructose converts into glucose in our bodies. So when you take in fructose, the first, your body metabolizes it and first turns it into glucose before it goes on with further reaction. And the last monosaccharide we're going to focus on is galactose. So galactose is an aldohexose. Once again, we know it's an aldo because here's the carbonyl group, and this carbon is bonded to a hydrogen and a carbon. It's a hexose. It's a hex because of the 6 carbons, and an ose because it's a sugar. So galactose is not found free in nature. We can get galactose and we can isolate it by hydrolyzing lactose, but we don't find it free in nature like we do glucose and fructose. We obtain it from lactose, so I've already said that. And lactose is a disaccharide, which I'm going to talk about in just a couple minutes here. Galactose has a similar structure to glucose, except that it has its a hydroxyl group on carbon 4. So 1, 2, 3, 4. This hydroxyl group on glucose is on the other side. So galactose and glucose are very similar in structure other than this hydroxyl group. You do not need to know that because once again I told you I'm not going to have you draw out these monosaccharides. So that last bullet there you can cross off when you're memorizing different facts about galactose. So cyclic structures are the prevalent form of monosaccharides with 5 or 6 carbon atoms. So it turns out in nature that if you have 5 or 6 carbon atoms, it tends to be more stable if it's formed into a ring. So cyclic structures form from the linear structures when the hydroxyl group on carbon 5 reacts with the carbonyl group on the aldehyde or the ketone. So I'm not going to have you memorize this particular reaction, but I do kind of want to take us through it. So here's carbon 5 because we count our carbons down from the top. So it turns out that the hydroxyl group on carbon 5 will loop around and react with the carbonyl carbon here. And then it's going to make a ring. So here's the oxygen on carbon 5, and it's reacting with that carbonyl carbon. And then it makes a ring. So upper level chemistry classes have to go through this reaction and draw it and talk about it, but I'm not going to have you do that. So, but what I do want you to be able to do is to be able to look at one of these cyclic structures and tell me whether it's in the alpha form or the beta form, okay? So both of these cyclic structures here, the one on the left and the one on the far right, is D-glucose. So when placed in solution, cyclic structures are going to open and close. So they're continually going through this reaction, which is why we have the forward arrow and the backward arrow. The alpha D-glucose is converted to beta D-glucose and vice versa. So here, if we look at this slide, here's our alpha D-glucose. And if we have a solution, we're going to find that we have about 36% of it in this form. And then it's going to open. And you're only going to have trace amounts of this because it's more stable if it's in a ring. So it'll open for a split second and then reform into a ring, and you're going to be left with about 64% of your cyclic glucoses being in the beta form. Okay, so you don't have to memorize these percentages or anything. I just wanted to give you an idea of the dynamics going on when you have one of these monosaccharides in a solution. So they basically just keep opening and closing. What I want you to be able to identify is right here. Let me grab my pen. Is focus on this chiral carbon right here. This is the carbon that used to be the carbonyl carbon. And the OH group that is on that carbon is what's going to decipher whether we're talking about the alpha form or the beta form. In this particular case, the OH group is pointing down. So that's where we're going to be getting our alpha. On this particular molecule, the OH group is pointing up, and so that's where we're going to get our beta. Technically, the next slide here, so I've already talked about that. It's a little bit more complicated to be able to find the alpha and beta than just simply down and up. The technical way of doing this is you find the carbon atom in the ring that has the two oxygens attached to it. So that was this carbon right here. When you find this carbon, there's an oxygen bonded here and an oxygen bonded down here. So that's going to be the important carbon you focus on. If that hydroxyl group on that carbon is trans, on the opposite side of the CH2OH found by going through the oxygen atom on the ring, then it's an alpha. So this is the technical way you do it. You find this carbon and you go through the oxygen to this carbon and you focus on this group. If this CH2OH is on the opposite side of the ring as this OH, then that's technically how we determine that it's an alpha. If we focus on this other one, here's the hydroxyl group. If I go through the oxygen to this carbon and I focus on this CH2OH, in this particular molecule, the OH and the CH2OH are both on the top of the ring. So since they're on the same side, we determine that that's beta. This is important in upper level chemistry classes because we actually write these cyclic structures flipped over and write them in a bunch of different forms. In this particular class, though, we're trying to keep it a little more simple. So the easier way to do it is going to be just to say if the OH group is on the bottom, it is alpha, and if the OH group is on the top, it's beta. One of the things that I heard a student say one time that I thought might be helpful, so I'm going to draw this here for you. Here's my waves on the ocean, and I am going to draw a little fishy in the sea. So here's my fish, and I am going to put a bird in the sky. And so a student once told me they had memorized that when the OH group is down, it's alpha because they think of the fish in the ocean. And when it's up high, they think of it being a beta because of the birds in the sky. Anyway, I don't know if that'll help you, but I'm going to keep the structures that same way, where that oxygen in the ring is going to be in the upper right-hand corner. So you can just do that down or up trick. Okay, so we're going to switch to 3 different disaccharides for you to focus on. A disaccharide consists of 2 monosaccharides. We are going to focus on maltose, lactose, and sucrose. So I want you to know those 3 disaccharides, and I want you to be able to tell me what monosaccharides make them up. So maltose is made up of a glucose and a glucose. Lactose is glucose and galactose. And sucrose, which is our table sugar, is made up of glucose and fructose. Okay. So a couple things to notice is they all have glucose in it. So really all you need to memorize is first they all have glucose, and then you just have to memorize the second one. What helps me is I can remember memorizing this as a student, and I love malts. So when my family goes to get shakes, I usually pay that little bit of extra to turn it into a malt. And somewhere when I was a student or just in life experience, I had known that glucose was a form of a sugar, and I love sugar. So I remember thinking, okay, I love sugar and I love malts. So malts are made up of just 2 glucoses. When I was memorizing lactose, I noticed that the word galactose has lactose in it. So lactose is made up of glucose and galactose. That was helpful. Sucrose, our table sugar, very sweet, and fructose is your sweetest monosaccharide. So that was helpful for me. Once again, when you hydrolyze a disaccharide, that means you're breaking it apart with water. Actually, this is the opposite. Never mind, we're not breaking it apart with water. This reaction is actually forming the disaccharide. So when two glucose molecules come together and make maltose, they're also going to take off, give you a water molecule. So hydrolysis, when you break apart with water, would be going the opposite direction. If we take maltose and we add water to it with some acid, you can break apart the maltose into glucose and galactose. So that would be the opposite of the reaction. So when you are taking water out, like the last way, we call that dehydration. So here is glucose and fructose. So pause for a second and tell me, what disaccharide am I making if I'm reacting glucose with fructose? And hopefully you said that you're going to make sucrose, because sucrose is made up of galactose and fructose. So when you put these two monosaccharides together, what happens in this dehydration reaction as it's making the disaccharide is water is roped off. So these two molecules come together and water is taken out of it, which is why it's called a dehydration. And then this oxygen here is going to bond to this carbon. So this carbon and this oxygen will bond together. And we get this bond. So that's dehydration, when you take off the water molecule. When you do hydrolysis, that means you take water and you're going to break the bond. And you are going to put water back in the molecule. So dehydration takes two monosaccharides and turns it into a disaccharide. If you hydrolysize, you're going to take water and you're going to break apart the disaccharide and get your monosaccharides back. So this is going through each of our disaccharides. I am never going to ask you to draw these structures or even draw this reaction, okay? I'm just going to take you through it and then I'll tell you what I want you to know. So here I'm putting glucose and glucose together. So I do hope that you can say, okay, glucose and glucose going through dehydration is going to make maltose. Because I told you I do want you to know what monosaccharides make up your disaccharides. So if I react a glucose and a glucose and I take off water, I am going to get maltose. And what we call this bond, this new bond here that forms where you have an oxygen between the two carbons, we call this a glycosidic linkage or a glycosidic bond. Okay? So what I do want you to know about maltose is this part right here, that your maltose is holding two glucose molecules together with an alpha glycosidic linkage or an alpha glycosidic bond. Okay? You're going to be able to recognize your alpha bonds because it's going to come down to the oxygen. Because remember, alpha meant that the OH group was down. If you think of that picture with the fish and the bird, alpha meant that it was down low. So this oxygen had been below this ring, which is why they keep that terminology alpha as being down. So maltose has an alpha bond holding it together. This is lactose. We know that because we can see it's being formed from galactose and glucose. Lactose happens to come from the beta form of the galactose. And so since beta has that OH group up high on the same side as our CH2OH, that's going to lead us to a glycosidic bond that they call beta. So this is what I want you to know from this side, that lactose has a beta glycosidic bond or link holding it together. And then I also want you to be able to look at this molecule and notice that it's a beta. So I might give you this disaccharide or the picture of maltose on the previous slide. I might give you that picture, and I'm never going to ask you to name it as either maltose or lactose, but I would want you to be able to look at this glycosidic bond and say, this is a beta, because it's not going down to the O, it's going up to the oxygen. So we get that kind of a straight line instead of the V. Here is sucrose, which is made from galactose and fructose. Once again, we're doing dehydration, so we're taking out water and we're going to bond this oxygen to this carbon to make our glycosidic link. This one is a little bit more complicated because the OH that is on glucose is reacting with the OH that is on fructose, and both of these have the alpha beta designation. So it turns out when you are making sucrose that you're going to end up with this both terms alpha beta glycosidic link, because the glucose is in the alpha form, the fructose is in the beta form, and it is that particular carbon that the reaction's happening with. So what do I want you to know from this slide? I want you to know that sucrose is a disaccharide. It is made up of glucose and fructose, and it has an alpha beta glycosidic link or bond. So here is a learning check. Identify the monosaccharides in each of the following. So lactose, I want you to memorize, is made from glucose. I'm having trouble finding my pen right here. It has glucose and it has galactose. I'm not necessarily super fond of this slide because we have the beta in here. When you are making, no wait, actually this isn't the one, I like this. So lactose is made from beta galactose and D-glucose. Maltose, a monosaccharide in maltose is made up of just glucose. So maltose is made up of two glucose molecules coming together. Sucrose is made up of glucose and fructose, and in this particular case the galactose has to be in alpha and the beta fructose has to be in beta. So if I was stuck with my circling trend. All right. The different sweetness of monosaccharides. What I have here is what they've done is they've taken some of the monosaccharides and the disaccharides, and here they've put on some artificial sweeteners. They took sucrose to be the reference point or the standard, and they said we're going to come up with these numbers to kind of relatively rate the sweetness of different sugars against each other. So they decided to make sucrose, give it the number 100, and then relate all the sweetness of the other things to sucrose, because remember sucrose is our table sugar. So I think we're all pretty familiar with how sweet sucrose tastes. So sucrose is just arbitrarily given this number 100, and then everything else is related relative to that. So you can look at maltose and lactose and see that they're not near as sweet as sucrose. Fructose, as I said, one of the sweetest carbohydrates found in nature, is sweeter than your table sugar. However, when you get down to these artificial sweeteners, which are not carbohydrates, they are artificially synthesized, these ones, look at how much sweeter they are than sucrose. This is why if you like putting saccharin in your drink, you're not going to put a whole heaping teaspoon in your tea or whatever drink you're having like you would sucrose, because it's just going to make it way too sweet. So sugars and artificial sweeteners, they differ in their sweetness. They are compared to sucrose, which is assigned the value of 100. So I've already talked about that. I'm not going to ask you any test questions on this. I just thought this was a pretty interesting information. There's a group of saccharides called oligosaccharides, and we're starting to hear more and more about these in literature that you read about, and the reactions and the health benefits we get from these in our diet. Oligosaccharides are carbohydrates made by condensing 3 to 10 monosaccharide units. So 3 to 10 monosaccharides gives us this new subgroup called oligosaccharides. They are found in legumes like peas and beans. You find them in onions, cabbage, broccoli, cauliflower, things like that. They increase the beneficial bacteria in the colon. So that's one of the good side effects of eating these oligosaccharides, is we're going to get more of those good bacteria in our colons. A downside to this, as we're all fairly aware of, is that it's going to increase gas production. You don't want to go on a car trip with somebody who's had a lot of beans. Another benefit of having these oligosaccharides in your diets is they help absorb the calcium ion from the intestines. They also help reduce cholesterol in the blood. So you're starting to hear more and more about the benefits of these. Here's a picture of just one oligosaccharide. This one is raffinose, And we have been showing you the structure of these rings as being these nice little hexagon shapes, but really in nature those 6 carbons in the ring, they bend into these different forms. So raffinose has 3 monosaccharides. This one here has a 6 carbon ring, or a 6 atom ring, because one of the atoms here is oxygen. This one has a 6 atom ring, and if you look over here, this guy here has a 5 carbon ring. I'm not going to ask you any questions about raffinose. I just kind of wanted you to see a picture of an oligosaccharide. All right, so switching gears, our last category. I've taken you through monosaccharides, disaccharides, and now I'm going to talk about polysaccharides. There's 4 polysaccharides we're going to focus on. Polysaccharides are polymers of D-glucose, so all of them are made up of D-glucose. They're going to include amylose and amylopectin, which are found in starches. Okay, so here's the first polysaccharide we're going to focus on, and here's the second one. We find them in starches. They're made of alpha D-glucose. And then we're going to focus on glycogen, which is also made from alpha D-glucose. And we find that in animal starch, or in, like, for example, muscles in animals. And then the last polysaccharide we're going to focus on is cellulose. Cellulose is found in plants and wood. It's usually found in, like, the plant walls. The thing that's different about cellulose is it's made from beta D-glucose. So if you look at the other 3, they are made from alpha D-glucose. So all of our polysaccharides are made from glucose. The first 3 are from alpha D-glucose, and then cellulose is made from beta D-glucose. So I want you to keep that straight. So going through each of these, starting off with amylose and amylopectin, we tend to find it in starch. So when you eat starchy foods, you're getting a lot of these 2 polysaccharides. To kind of focus on the difference between amylose and amylopectin, here amylose is just a straight chain. It doesn't have any branches coming off of it. So this one right here is showing us our amylose, and it's got the D-glucoses all bonded together. I would hope that you could look at this after studying, look at this and say, oh, this is an alpha glycosidic link because my oxygens are going down. So it's making that V shape. That's going to be familiar for your alpha glycosidic bonds. In this particular case, the alpha bond is happening between carbon 1 on one ring and carbon 4 on a different ring. So I do want you to know that in amylose, the bond holding it together is the alpha 1,4 glycosidic bonds. I have this on a later slide, so you don't have to write that down. When we switch to amylopectin, it's going to look very similar to amylose. It's going to have your straight chain here of alpha D-glucoses bonded together. But what it's going to have different is it's going to have these branches. And the branch on the amylopectin is going to be between carbon 1 on one molecule and then carbon 6 on the main chain. So this one's a little more complicated. So amylopectin, I do want you to know, is held together by alpha 1,4 glycosidic bonds, and then the branches occur at an alpha 1,6 location. All right, and then this picture is kind of showing how they spiral on each other. So if you were to have a potato, it turns out that about 20% of it is amylose, the straight chain polysaccharide, and about 80% of it is amylopectin, which has all the branches on it. I don't expect you to memorize those percentages. So a little bit more in detail. Amylose, it is a polymer of alpha D-glucose molecules. So here they are. They are linked together by alpha 1,4 glycosidic bonds, and they are a continuous unbranched chain. So I want you to memorize these 3 points. I want you to know these same points on all of your polysaccharides. I want you to know what monosaccharides they're made from, what kind of link is holding them together, and whether it's branched or unbranched. So amylopectin, also made from alpha D-glucose. It is a branched chain, and it has alpha 1,4 glycosidic bonds holding your main chain together, and it has alpha 1,6 bonds at the branches. Okay, so one more time. This is carbon 1 on one of the molecules, and this happens to be carbon 4. Because when we count these in the ring, carbon 1 is always that special carbon. that has the 2 oxygens bonded to it, and then you count clockwise. So that's carbon 1, this is 2, this is 3, and this happens to be carbon 4. So I'm not going to ask you to draw that. This is just kind of a memorizing point. Okay, the third polysaccharide is glycogen. It is found, oh, it's not going to where it's found yet. It is a polysaccharide that stores alpha-D glucose in the muscle. So this is one of the storage places for glucose. So if you are not getting enough in your diet, your body's going to start breaking down your muscles to start hydrolyzing it to access these glucose molecules. So glycogen is stored in, it says here in muscles. Sometimes the test questions will say that it's stored in animal cells versus your amylose and amylopectin, which is stored in plants. Glycogen is very similar to amylopectin, but it's more highly branched. So all those bullet points I had for you on amylopectin are going to be the same for glycogen. It's going to have alpha 1,4 glycosidic bonds holding the main chain together. It's going to have alpha 1,6 bonds holding the branches. The 2 differences between glycogen and amylopectin is where their source is, where they're found. Amylopectin is in plants, glycogen is in animals. And the other difference is that glycogen is more highly branched. So I have this picture here to kind of drive that point home. Here's amylopectin with some few branches coming off, and then glycogen with a lot of branches. All right, our last polysaccharide we're going to focus on is cellulose. It is a polysaccharide of glucose, like all of our polysaccharides. However, this one has beta bonds. Okay, that's the difference. It is unbranched, so it's similar to amylose, because amylose is our unbranched polysaccharide, and cellulose is unbranched. It is held together by beta 1,4 glycosidic bonds. Cellulose is the only one that has the beta bonds. And we notice the beta bonds because if we look at the structure, the oxygen is up. It is on the same site as our CH2OH. Cellulose cannot be digested by animals, by humans, because we don't have the enzyme that can break down a beta 1,4 glycosidic bond. So that's why you tend to call your cellulose the roughage in your diet, because we don't metabolize it. They're showing a picture of cotton here because cotton is a plant that has a very high percentage of cellulose. Cellulose is also found in like the plant walls, the cell walls. So they tend to give your plant the stiffness on the outside of those cell walls. So here's a learning check. Identify the polysaccharide and the type of glycosidic bond in each of the following. So we've got our pictures. We just talked about the cotton, so I'd have to somehow, if this was a test question, identify you into the fact that we're focusing on the plant structure, not the plant starch. Okay, if we're talking about plant starch, then we're over here on picture B. So this would be our plant structure or the cell walls. So identify the polysaccharide. So this would be cellulose. Identify the glycosidic bonds. Okay, so cellulose has got only beta 1,4 glycosidic bonds. Focusing on the potato, so they want us to think of the plant starch. Okay, so you might want to label this picture as plant starch because I'm not going to be giving you pictures on your exam. This one has amylose and amylopectin. And amylose has alpha 1,4 glycosidic bonds. Amylopectin has alpha 1,4 and also alpha 1,6 glycosidic bonds. Over here we have muscles, or sometimes they call it animal starch. Different from plant starch. And this is going to be glycogen. And glycogen has the exact same bonds that amylopectin has.